And you will do that for different points. We went from 5,000 cells to 500,000 cells. And then we've got a kind of calibration curves. I took an example for the microcystis genus. We finally, in generation 3, had 3 probes specific for this genus. And we extracted 5 points corresponding to 5,000, 25,000, 100,000, 250,000, and 500,000 cells, for each cell corresponding to an amount of RNA. And this amount of RNA hybridized to the microarray gave a signal-to-noise ratio. It means a fluorescence signal. And from these curves, you can draw some average line and got an equation. And when your square R is close to 1, it means that your equation is more or less perfect, and you can calculate the cell number from the signal-to-noise ratio. Then you can transform your fluorescence into a cell number. So what we did with the calibration number and the generation 3 microarray of microaqua, we field tested the microarray in different kind of water. So I'm describing now the panel 3 results. So the first site was a freshwater site. So this microarray was designed for freshwater, but we choose to test it on the marine water as a negative internal control, and on brackish water. This lagoon was quite particular because it was very eutrophied and always very full and of different kind of cells over season. We collected 51 samples over a 2-year period. What did we do with these samples? We extracted all the RNA of these samples. We extracted the DNA. We also measured the number of cells per liter of cyanobacteria and of bacteria. And we did that for the 3 different water sampling sites. We use the RNA that we fluorescently labeled with the 5,5 fluorophore. We analyze the result like Dr. Weigel told you, and then we obtain some data of the fluorescence signal and transform the fluorescing signals in a graph to compare all the samples together. So here I put a comparison of the microarray result over the 3 different sites, the brackish water, the marine water, and the fresh water, to show you that the behavior of the microarray is very different depending on the type of water. As I told you, this water was really rich in microorganisms. So you've got the eubacteria probe, which is a general probe which recognize all the bacteria, gives a very high signal. The marine water gives a lower signal. We are talking about RNA, so it's related to activity. We're not talking about DNA. So marine water are not very rich in microorganisms, active microorganisms. And we've got the fresh water, which responded really well to the eubacterial probe. Then when we're doing the phylum bacteria, which is a Yarki probe, which is a little bit under, we've got the same kind of signal, which were quite high. But when we're looking at one of our targets, we can see that the target was not present all the time, and certainly not in the marine water, which is normal because we usually don't find this kind of microorganism in marine waters. The next step was to transform all this fluorescence signal into cell number, because that was the aim of the project. So we used the calibration curves, and we draw for each of the calibration curves the corresponding curve of cell concentration. And you see that according to the probes, you've got a range of cell estimation. Why this is quite high? is because the square R is not perfect. It's far from 1. So we trust in more the curves, the cell number curves, which are below. And then this transformation of results can give you a range of cells per liter. Then you are able to follow seasonally the cycle, the life cycle of these microorganisms. Now we've got a calibration curve. We've got a transformation from the fluorescent signal to a cell number, but how can we be sure that the cell number is a true cell number? The next step of validation was to use some alternative methods to check that the cell number we saw with the microarray was the same cell number we saw with alternative method. So here I introduce you the qPCR that was designed to detect a bacteria, a family of bacteria which is called Vibrio. And this is the microarray result for the 3 different water sites, the brackish, marine, and freshwater. And you see that the PCR count gives exactly the same trend as the fluorescent signal of the microarray. This was a very good news for us, because it means that the microarray behaved like a traditional approach. But we were not satisfied enough, so we changed and we used another approach to quantify the cells. So I'm presenting you samples from a Netherlands partner. We who counted with microscopy alternative method the number of microcystis present in the sample he sent us. In our side, we did a microarray analysis. Here you've got the sample count, and here you've got the microarray analysis. And we were quite happy because we think it's not too bad. We are in the same range of cell quantification. So we've got another approach, traditional method of microscopic counts with microarray, which shows very coherent results. We also did another verification. We just studied the Anabaena genus, and we've got the microarray results. And here, the toxin variant analysis of by mass spec LMS. And we see that the presence of this genus is quite well correlated with the presence of the toxin. So it's another way to show that the fact we see the organisms is correlated with the presence of the toxins. And I will introduce you a last case study, which involves another European project, MicroCockit. This project used the microarray to evaluate the presence for the target we have on the microarray. And then they collect some samples from different sites. They send us the RNA they extracted, and we did the microarray analysis. So what we see is the evolution of the profile according to the site. And what they're going to do, they will take the same samples and do pyrosequencing. Then we will have a correlation between the sequencing data with our microarray results. So it will be a third approach to confirm that the microarray is really reliable. So I'm talking a microarray about 20 minutes now, but why I would like to try to convince you to use microarray? Because it's a very quick method. Now you've got pyrosequencing. It's a huge amount of sequence available to know the assemblage of your bacterial population. It gives you a proportion of what's present in your sample, but you need about 3 months to get your data analyzed from the moment you extract your sample until the moment you get your result. With a microarray, you've got your result within 3 hours, and you target only the microorganisms you are interested in. The second approach, which is the classical approach for most of microorganisms, like microalgae or cyanobacteria, is microscopic counting. So for this method, you need time. It's really time consuming because you have to check visually your sample, count each species present in your sample, so you need time, but you need also a taxonomy expertise. You have also the hybridization, which is fish, fluorescent situ hybridization, but you can't hybridize more than 2 probes on the same sample because you are limited by the dye you're using. You've got the qPCR, but until up to now, I don't know more than 8 multiplex species together. And you've got the problem of enzymatic inhibition. When you're doing PCR, all the system is based on enzyme. And environment, you don't master the condition of your environmental samples, and usually it's not pure enough to cope with enzymatic inhibition of PCR. The advantage of microarray, when you think about the cost of all these methods, on microarray you can have more than 50 different species spotted on your microarray, and there's no limit in probe spotting. It's just the only limit is the database you have to design the probe, and the whole technology costs around 50 euro. So for 50 euro per sample, you can detect whatever microorganism you want, since you've got the right probe design. So it's not fairy tales, because there's some constraint in the microarray technology. First, you find only what you look for. Second, your knowledge is limited by the database, because we don't know everything about diversity, microbial diversity. Maybe we know about 2–5% of what exists on Earth, and there are some possibilities that there's some cross-reaction with an organism which is not identified yet, even if the database is increasing very quickly every day. We have quantitative limits, because if your target is in very low concentration level, you can't detect it. You've got genetics limits, because microorganisms evolve more quickly than humans, and after a couple of generations you can have spontaneous mutation, and if the mutation is within the probe design, your microarray won't work anymore. And it's highly dependent on the database. But there's some solution to all this issue. What we usually do when you're designing a microarray, you're checking that with biosequencing, because biosequencing can generate a lot of data. Then you are able to check who's here, and if the genetic base of your macroarray is well done. Then you can design your probe set with this data in a better way, in a more efficient way. And you can expect the genetic drift. If you do biosequencing regularly, you can see if there's some mutation on your target organism you're chasing for. You've got similar quantitative limits for the biosequencing and qPCR as the macroarray, because they're all molecular tools. So they're all the same constraint that you're looking for, what you want to find. And there's some inhibition problems with the PCR approach. The advantage of macroarray is that if you want to design a new probe for a new target, it's not a big work, and you can add it to your macroarray and implement it with something else. You can put as many probes as you want on the macroarray. There's no limit. It's quite fast, it's reproducible, and very sensitive. So I think it's a really good technology to do water monitoring. And as we've got an implementation of the gene database every day, we're reaching billions sequencing in the database. So even if we don't know every microbial diversity, we have more problem to treat and analyze the implementation of the gene database than to using it. So there's always solution to issues, just have to take time to find the right one. Thank you for your attention.